Rakhigarhi, Haryana, India, 2015. Dr. Vagesh Narasimhan at Harvard Medical School, working alongside Dr. David Reich and in direct collaboration with Dr. Vasant Shinde of Deccan College in Pune, published the results simultaneously in Cell and Science in September 2019. The Rakhigarhi genome, individual I61war3, a woman from the Indus Valley Civilization, contained zero ancestry from steppe pastoralists, not trace amounts, zero. The dominant paternal marker in North Indian Brahmin communities today, haplogroup R1AZ93, present in 40 to 70% of their male lineages, was completely absent from the largest Bronze Age city on Earth. But here's where it gets strange. The people who built those cities, the first urban sewage systems predating Rome by 2,000 years, the trade networks reaching deep into Mesopotamia, were genetically different from the population that would eventually claim their land. The question is not who they were. The question is what happened to them. Here is where this episode ends. Roughly 1900 years ago, around 100 CE, the genetic mixing of India's ancestral populations came to a near-total halt. Not because of a war, not a famine, not a migration, because of a social institution. The caste system did not merely organize Indian society. DNA confirms it, genomically sealed it. And the consequences of that sealing are still measurable in the bodies of South Asians alive today. That is the destination. What follows is how we got there. If you've ever actually wondered where you come from, not the family story, but the real biological answer, then subscribe, because this goes deeper than most people are willing to go. Because the deepest layer of South Asian ancestry is not the Indus Valley Civilization. It is not the arrival of steppe pastoralists. It is something far older, and its closest living relatives are not in India at all. They are on an island their descendants have never visited. Before we go further, if you've ever taken an Ancestry DNA test, did your results surprise you? And if you're South Asian, did the numbers match your family story, or did they tell a completely different one? Drop it in the comments, because what you're about to hear may change how you read whatever was on that report. The Andaman Islands, Bay of Bengal, right now, today. On Little Andaman Island, a community called the Onge live in conditions of partial self-chosen isolation. There are fewer than 100 of them. Their language is unrelated to any other living tongue on Earth, and in their genomes, the closest living approximation of a population that arrived on the Indian subcontinent 50,000 years ago is the genetic foundation of every human being who has ever called South Asia home. Most South Asians have never heard of them. Most South Asians carry their DNA. This founding population has a name in the scientific literature: Asi, ancient ancestral South Indians. The lineage diverged from a wider East Eurasian metapopulation somewhere around 40 to 50,000 years before the present. Narasimhan, Patterson, and Reich's 2019 science study confirmed what earlier analyses had suggested. The Onge of the Andaman Islands serve as the closest genomic proxy we have for these first settlers. And the distribution of this ancestry across the subcontinent today is anything but uniform. Among the Irula tribal communities in Tamil Nadu and the Paniya communities in Kerala, AC ancestry exceeds 70 to 80% of total genomic composition. Among North Indian upper caste populations, that same lineage drops to 25 to 35%. But here is the thing. It is present in every single South Asian genome without exception. The Onge ancestry is inside every person whose family ever came from the subcontinent. The only question is how much. This discovery was not made yesterday. The evidence has been building for years. Nobody talks about it enough, until now. Dr. Neeraj Rai, who established the ancient DNA laboratory at the Birbal Sahni Institute of Paleosciences in Lucknow, India, prepared the Rakhigarhi samples for sequencing. What his team found in the genome's non-West Eurasian component pointed directly toward the Andamanese. A woman buried 4,500 years ago in Haryana carried genetic signals that trace back to islands her civilization had no contact with, because both of them descended from the same people who crossed into South Asia before agriculture, before pottery, before there was a word for anything. Think about that for a second. The gap between the oldest Asi settlers and the Rakhigarhi woman is longer than the gap between Rakhigarhi and you. The deepest layer of South Asian ancestry is older than humanity's first art, older than our first known burial, older than symbolic behavior as archaeologists can currently trace it, and it is in you. The standard narrative of Indian population genetics begins with the Indus Valley Civilization, but the IVC sits on top of something vastly older, and that older substrate is not background noise. It is the dominant ancestry signal in over 400 million living South Asians. Here's what nobody in the popular conversation about Indian origins has fully reckoned with. The distribution of IC ancestry across the subcontinent is not random. It maps, with uncomfortable precision, onto caste position, language family, and geographic latitude. That distribution was created by a specific sequence of migrations, and then enforced by something else entirely. But before we get to the enforcement, there is the question of who arrived next. And the answer to that question rewrites where South Asian farming came from. The Iranian farmer-related ancestry present in the Rakhi-Garhi genome split from the actual Iranian farmer lineages more than 12,000 years ago, placing it as an independent population on the Iranian plateau long before any Fertile Crescent farming expansion. The first farmers of South Asia were not immigrants from the West. They were already there, Mohenjo-Daro, Sindh, Pakistan. Approximately 2600 BCE. A city of 40,000 people. Streets on a grid. A drainage system carrying wastewater through brick channels beneath the roads. A technology. Rome would not independently develop for another 2,400 years. Merchants here used standardized bronze weights, measured in precise binary fractions. Their trade goods have been found in Mesopotamian graves. Their script, inscribed on over 4,000 objects, has never been deciphered, and the people who built it were not brought here from anywhere else. The standard model of how civilization spread assumes the Fertile Crescent exported farming eastward into South Asia, the same way it exported it westward into Europe. That model is wrong. The Rakhi-Garhi genome shows that the split between the IVC's Iranian-related ancestry and the actual Fertile Crescent lineages predates farming itself by thousands of years. The IVC was not downstream of Mesopotamian or Iranian influence. It was a parallel development, an independent invention, a civilization that grew from within. And yet, the data said something even stranger. Every model that frames South Asian civilization as a recipient of cultural or biological waves from the West requires revision. The Harappan people were not recipients. They were a distinct, locally developed population who created urban complexity on their own timeline, from their own genetic stock, with their own script, their own weights, their own drains. The parallel to the debate over Indo-European language origins is not incidental. It is the precise center of everything that comes next in this story. The single piece of evidence underpinning all of this, the entire genomic case for the independent origins of Harappan civilization, rests on the DNA extracted from one inner ear fragment, one petrous bone, one woman, buried with a necklace of gold beads in a city that no longer exists. In a script no one can read, speaking a language we will probably never recover. 61 skeletal samples were screened from Rakhi-Garhi. Only one yielded usable ancient DNA. Over 100 sequencing runs were pooled, and from that near-impossible recovery came the confirmation. The people who built this place were indigenous. They were not migrants who could retrace a path. They were the origin point, which makes what happened to them not a question of source populations. It is a question of survivors. Because the direct genetic descendants of the Indus Valley Civilization, the closest living populations to the people who built Mohenjo-Daro, are not in the cities of India today. They are in the forests of Tamil Nadu and Kerala, in tribal communities carrying genomic signatures that mark them as the civilization's closest biological heirs. They have no memory of cities. Northern Pakistan, 2023. A cave in the foothills of the Himalayas. A research team extracts a stalagmite from the cave floor, a formation of calcium carbonate that has been recording rainfall data in its isotopic layers since before the Indus Valley Civilization existed. When they read the oxygen isotope ratios locked inside it, they find 4 separate droughts, each one lasting longer than a human lifetime. The longest stretches 164 years. It peaked around 1733 BCE. The IVC's urban heartland was already emptying when it began. A study published in Communications Earth and Environment traced these drought periods across a span from roughly 4,400 to 3,400 years ago. Annual rainfall declined 10 to 20%. Average annual temperature climbed by half a degree, not in a season, not in a decade, over generations, and across that slow, relentless drying, 4 extended drought periods arrived, each one longer than a human lifetime, and each one pushed the IVC population further from the river systems their civilization depended on. Consider what 164 years of drought actually means. You are born into it. Your parents were born into it. Their parents were born into it. The drought is not an event in your lifetime. It is the condition of your existence. And you have 5 million people trying to find water. The IVC did not collapse. It migrated. Ancient DNA from the Swat Valley, sequenced as part of the Narasimhan 2019 science study, shows IVC-related genetic ancestry appearing in highland settlements as the urban centers emptied. The population dispersed southeast and northeast along river systems as the monsoon pattern failed. The civilization fractured into the ancestral populations that would eventually mix with the incoming steppe migrants. The people of Mohenjo-Daro became the substrate for everything that followed. The final occupation layers of Mohenjo-Daro contained fewer standardized goods, simpler pottery, and skeletal evidence of individuals who died in the streets and were not buried, not killed in battle, not executed, simply left. The people who made the weights and built the drains and traded with Mesopotamia did not leave a record of being conquered. They left a record of slowly stopping. In our last video, we looked at what soldiers at Agincourt were doing chemically to their bodies just to survive combat. Different civilization, different millennium, same pattern, a population under impossible pressure, finding ways to continue. The people of the IVC had no army. They had only the direction of the rivers. Drop in the comments which collapse hits harder for you, the sudden one or the slow one. Back to the DNA. Because the slow collapse of the IVC is precisely what made the arrival of the steppe population possible, and the genetic signature of that arrival is something you may be carrying right now. What the collapse created was a demographic opening in the northwest of the subcontinent, a depopulated corridor through which, within a few centuries, an entirely different population would arrive from the steppe. The drought did not end the IVC's genetic contribution to South Asia. It repositioned the IVC's survivors. It moved them out of the path of the migration that was coming. And the migration that came was primarily, documentably, a movement of men. Swat Valley, northern Pakistan, approximately 1200 BCE, a burial site. Bodies interred in what archaeologists would later classify as the Swat proto-historic grave-type culture. The individuals here were analyzed as part of the Narasimhan 2019 dataset. For the first time in South Asian ancient DNA, they carry a detectable steppe ancestry signal, perhaps 10 to 20% of the autosomal genome, so the steppe migration had arrived. But the Y chromosome tells a different story. Only 5% of the Y chromosomes recovered from these burials carry the R1A-Z93 subclade that defines the central steppe Middle and Late Bronze age population. What that means is this: the steppe ancestry detected in these early South Asian individuals came primarily through women. Steppe women mating with local IVC-descended men. That is what the early signal shows. Then, several centuries later, in present-day South Asians, the pattern reverses completely. Narasimhan et al. Directly analyzed this asymmetry. In modern South Asians, there is a documented excess of steppe ancestry on the Y chromosome compared to the autosomes, a statistical Z-score of 2.7 for an excess from males. That reversal between the early Swat Valley data and the modern genetic landscape documents a shift in the direction of the mixing. Steppe women arrived first. Steppe male lineages came to dominate the paternal record afterward. What they found next changed everything they thought they knew about this. The mitochondrial DNA of modern South Asian women shows very little steppe influence. The Y chromosome of North Indian Brahmin men shows 40 to 70% steppe-related haplogroup R1A-Z93. That asymmetry is not a statistical artifact. It is a record. The ancient DNA does not record the social mechanism, whether this was intermarriage, elite male replacement through prestige and hierarchy, or something more coercive. What it records is the outcome. A demographic event that preferentially altered the paternal heritage of the subcontinent's northern populations and left the maternal line almost entirely intact. Among the Kalash people of northern Pakistan, a small, isolated community in the Hindu Kush Mountains who speak an Indo-European language and have preserved ancient ritual practices, R1A-Z93 reaches exceptionally high frequencies. Their paternal lineage is among the most steppe-proximate in all of South Asia. They are 3,000 years from the migration. They are still primarily its men. The sex bias of the steppe migration is the specific mechanism through which the genetic geography of the subcontinent was set up. The paternal line transformation of northern populations with steppe-derived Y chromosomes, while local maternal lines largely survived, created a landscape primed for the stratification that would follow. The genome was already being organized by hierarchy before hierarchy became doctrine. And then something happened that the models did not predict. The 1900 years of mixing that followed the steppe migration, the period between roughly 2200 BCE and 100 CE when the ancestral populations of South Asia blended, should have continued indefinitely. By every genetic model, the subcontinent was on course to become one of the most thoroughly admixed populations on Earth. Then it stopped, completely. And the reason it stopped looks like a wall. Before we go any further, it is intellectually important to take the strongest possible version of the opposing argument seriously. And on this topic, the opposing argument has real teeth. The out-of-India theory, the hypothesis that Indo-European languages originated in the Indian subcontinent and spread outward towards Central Asia and Europe rather than arriving from the steppe, is not a position held only by nationalists or amateurs. It is held by serious scholars who point to real evidence. The exceptional diversity of R1A subclades found in South Asia, the deep roots of Vedic tradition in the subcontinent. The apparent paradox that the homeland of the world's most widely spoken language family shows, by some measures, more R1A diversity than regions that supposedly received it. Dr. Peter Underhill and colleagues at Stanford published phylogeographic analysis of R1A in 2015 in the European Journal of Human Genetics. Analysis that was subsequently cited in both directions, its data forming part of both the steppe origin and South Asian origin arguments depending on which assumptions were applied. That is not cherry-picking. That is genuine scientific ambiguity. The coalescence age of R1A Z93 has been estimated at approximately 4,000 to 4,500 years ago. Some researchers argue this is consistent with a South Asian origin, an expansion outward towards Central Asia preceding the inward migrations the steppe theory requires. The case in its strongest form is this. If the diversity is in India, perhaps India is where it started. Hold on to everything I just told you, because it's about to mean something completely different. Here is why the ancient DNA cannot be reconciled with this model, regardless of how the modern diversity data is interpreted. The out-of-India theory makes a specific, testable prediction. If Indo-European languages originated in South Asia and spread outward, then ancient DNA from steppe populations prior to 2000 BCE should show South Asian genetic ancestry. It does not. The IVC genome at Rakhigarhi, dated to approximately 2500 BCE, contains zero steppe ancestry, which is exactly what the steppe origin model predicts, and exactly what the out-of-India theory cannot accommodate. More critically, the Sintashta culture, dated 2100 to 1800 BCE in what is now Russia and Kazakhstan, the archaeological culture directly ancestral to the migration that produced steppe ancestry in South Asian ancient DNA, shows no South Asian ancestry at all. The out-of-India theory requires a population moving from India to the steppe without leaving a single detectable genomic trace in any ancient sample collected from the steppe. That is not a gap in the data, that is a falsification. In 2018, before the Rakhigarhi results were published, the controversy around what the genome might show had become politically charged enough in India that the publication timeline reportedly faced pressure. Dr. Niraj, whose laboratory was doing the sequencing in Lucknow, stated publicly that the individual showed no steppe ancestry. The data existed in a state of political sensitivity before it existed in a published journal. Science was ready. The climate around it was not. The defeat of the out-of-India theory by ancient DNA does not diminish the Harappan civilization. If anything, it makes the IVC more remarkable, a civilization that grew independently, without the migration wave that supposedly gave civilization to everywhere else, only to have its territory eventually carry genetic and linguistic traditions from entirely elsewhere. The cultural achievements of the IVC are confirmed as genuinely indigenous, and the languages spoken by the people who built those achievements were almost certainly not the ancestors of Sanskrit. But here is what the ancient DNA has done that nobody fully anticipated. It has not resolved the culture war. It has given both sides sharper instruments. The genetic fault line created by the steppe migration, the gradient between high steppe North Indian populations and low steppe South Indian populations, running like a ridge across the subcontinent, maps with striking precision onto contemporary linguistic, social, and political divisions that are today explicitly framed as contests over whose version of Indian history is true. The genome does not take sides, but it does keep the score. Chennai, Tamil Nadu, 2024. A graduate student receives her commercial ancestry test results. The report shows approximately 65% ASI-related ancestry and 8% steppe-related ancestry. Her paternal surname traces to a Brahmin lineage. She is confused. She expected higher steppe numbers, given her caste background. What she is looking at is the South Indian exception, a region where the mixing of ancestral populations happened earlier, more completely, and at a higher ratio of indigenous to steppe ancestry than anywhere in the north. Her genome reflects a specific regional history that cannot be explained by caste label alone. That number is not random. It is a position on a map. Dr. Priya Morjani and colleagues, working with Dr. David Reich at Harvard Medical School and Dr. Kumarasamy Thangaraj at Shir CMC in Hyderabad, published genome-wide data from 73 Indian population groups in 2013 in the American Journal of Human Genetics. They analyzed linkage disequilibrium, the way ancestry segments break up over generations, to estimate when the mixing of ancestral North Indians and ancestral South Indians actually happened. The answer ranged from approximately 4,200 to 1,900 years ago, varying by group. Northern populations show more recent admixture dates consistent with multiple incoming waves. Southern populations show older dates, consistent with earlier mixing that was not subsequently reinforced by the steppe migration. This isn't just ancient history. The gradient is in your DNA right now. Anyone with South Asian ancestry who has ever taken a commercial test and seen their ancestry percentages is looking at a specific position on this gradient. The steppe number reflects where their northern ancestors were standing when the Bronze Age migration arrived. The AC number reflects how much of the pre-migration population survived into their lineage. And the Iranian farmer-related number reflects the IVC. Dr. Kumarasamy Thangaraj, one of India's foremost population geneticists and a co-author on both the Morjani 2013 study and multiple South Asian ancient DNA analyses, has noted in published work that the genetic structure of Indian populations is unique globally. Thousands of groups with distinct genetic profiles, each shaped by millennia of isolation, creating a natural laboratory for population genetics that exists nowhere else on Earth. The laboratory was not designed by scientists. It was designed by a social system. The standard framing treats South Asian genetic diversity as simply the natural result of a large, complex population. Ancient DNA reframes it as a sequence of specific datable events, a steppe migration with a documented sex bias, followed by a period of active widespread mixing, followed by something that stopped everything. The current genetic landscape of the subcontinent is not the product of gradual, continuous flow. It is the product of a sequence, and the last event in that sequence was the most consequential of all, because here is what the admixture dating shows. The mixing of ANI and ASI happened across India between roughly 4200 and 1900 years ago. Then, for most population groups, it stopped, not gradually, not slowly. The genomic signal shows a cessation that, across most of the subcontinent, is consistent with having occurred within a relatively narrow window around 100 CE, and the genetic diseases that appear at elevated frequency in specific South Asian communities, certain cardiomyopathies, hereditary hearing loss, specific metabolic disorders, follow the exact boundaries of endogamous castes. They are not ancient misfortune. They are the direct biological result of a decision made approximately 1900 years ago, and they are being diagnosed in hospitals today. A cardiology ward, India, present day. A patient, 44 years old, South Indian Brahmin lineage, no family history of cardiac disease, is diagnosed with a specific form of hypertrophic cardiomyopathy caused by a variant with elevated frequency in their subcaste population. The variant is rare globally. In this jati, the specific endogamous community, it appears at a frequency 12 to 15 times higher than in the general population. The genetic counselor explains, this is a founder variant. It became elevated in frequency because, for approximately 1900 years, no genetic material from outside this community entered it. 1900 years, roughly 50 to 100 generations. Dr. Priya Morjani and colleagues found that for most Indian groups, 100% of ANI-AACE admixture is consistent with having occurred before approximately 100 CE. After that point, the admixture linkage disequilibrium data shows no further mixing signal in most populations. A 2025 cell study analyzing 2,762 whole genomes from 85 Indian population groups, spanning roughly 50,000 years of South Asian genetic history, confirmed the scale of endogamic isolation. Thousands of genetically distinct populations, each shaped by founder effects consistent with strict endogamy running approximately 50 to 100 generations deep. There are three things this tells us. The third one is the one that changes everything. First, the caste system did not merely organize Indian society. It genomically froze it. What 40,000 years of natural population movement, 3 massive migration events, 2 ancestral mixing periods, and 1 Bronze Age civilizational collapse had failed to do, fully separate the genetic destinies of South Asian populations. A social institution accomplished in a few generations, around 100 CE, with a consistency so total that its signature is readable in a modern hospital's genetic testing results. Second, the caste system is not only a social reality, it is a biological one, and the biological consequences are not distributed equally. Third, Dr. David Reich, writing in Who We Are and How We Got Here, noted that the endogamy practiced in India is so extreme and so ancient that many Indian population groups are more genetically isolated from their geographic neighbors than Europeans are from East Asians. 2 people from different Indian jatis, living in the same city, attending the same university, can be more genetically distant from each other than a Norwegian is from a Japanese person. That is not hyperbole, that is a measurement. You already know the name of the institution that made this measurement possible. The genetic diseases concentrated within specific South Asian castes are not the result of ancient migration or ancient environment. They are the result of a social structure that prevented the genetic dilution that would otherwise have occurred. Every South Asian person who carries a founder effect variant carries in their genome the biological record of a decision. The lock exists in the DNA. The key has never been found, but here is what the ancient DNA cannot do, what no genome, no matter how carefully sequenced, can ever show. Beneath the stratification, beneath the endogamy, beneath the step gradient and the caste boundaries and the linguistic divides that run across the subcontinent like fault lines, the Rakhigarhi woman is the ancestor of all of them. IVC Klein ancestry is present in every single South Asian population that has been analyzed, from Brahmin communities in Kashmir to Dalit communities in Tamil Nadu. The lock held for 1900 years. But the deepest layer, the one that predates the step, predates the IVC, predates everything we call Indian history, could not be locked. It is in all of them, the same deepest origin, the same petrous bone, the same AZ signal pointing back to an island in the Bay of Bengal that South Asians have never thought about. Rakhigarhi, again, one petrous bone, one woman, gold bead jewelry, dead for 4,500 years. Her genome contains no step ancestry. She predates the migration by 600 years. She predates the caste system by 2,400 years. She is the genetic common ancestor of populations that today occupy radically different positions in a social hierarchy so old that many of its members believe it has always existed. Her DNA was recovered from fragments of her inner ear after 100 failed attempts, and it is inside the bodies of people who have no idea she existed. A 2025 cell study, the most comprehensive whole genome dataset assembled for South Asia to date, covering 85 Indian population groups across roughly 50,000 years of genetic history, confirmed one finding. Above all others, IVC Klein ancestry is present in every single sampled South Asian population without exception, from communities in the far north to communities in the deep south, across every caste level, across every language family. The Rakhigarhi woman's population is inside all of them. If you have South Asian ancestry, any South Asian ancestry from any caste, any language group, any region, you carry that signal. Not metaphorically, measurably. The same admixture modeling that confirmed her ancestry's presence in the 2019 dataset has since confirmed it in every modern South Asian population. Analyzed, the step arrived and stratified. The IVC collapsed and persisted, the lock held for 1900 years. But the deepest layer could not be locked. Dr. Lalji Singh, co-senior author on the Morjani 2013 study, one of the founding figures of South Asian population genetics, formerly of CSIR Sisambi Hyderabad, said after the admixture dating results were published, the fact that every population in India evolved from randomly mixed populations suggests that social classifications like the caste system are not likely to have existed in the same way before the mixture. He died in 2013, before the Rakhigarhi genome was published. Before the oldest face of the population he spent his life studying was finally recovered from the mineral density of an inner ear. He never got to see her. If this episode changed how you understand South Asian ancestry, subscribe and share it with one person who should see it. The next episode goes deeper into this story, and it is not one that has been told before. She was buried with gold. Her civilization did not survive. Her genome did, and the system built on top of it. The hierarchy, the endogamy, the lock, is 1900 years old. The DNA beneath it is 50,000.